Okay, well, welcome to today's lecture. We're going to talk about cell surface markers. So there are a lot of different cell surface markers, and my goal is to cover what they are. So, for example, when somebody says CD3, what are they talking about? When somebody says CD8 positive, what does that mean? So that's my goal today, is to try and figure out what these cell surface markers, and trust me, there are tons. I'm just going to cover some of the basics, some of the higher yield stuff that you'll see more often, some of the stuff that you could be tested on. Depending on what level you're at, I am just going to go over the very basics. So I am aware that I'm leaving out a lot of material because there is a lot out there. So now let's start talking about some of the different markers. So someone says CD3. So if a cell is CD3 positive, it means they express the CD3 cell surface marker. Well, what does a CD3 cell surface marker mean? That simply means that this cell is a T cell. So CD3 positive cells are going to be T cells. And this is going to be an important part to remember. So not B cells, T cells. And there are two different types. You have the cytotoxic T cell and then the T helper cell. So T helper and the cytotoxic. Alright, so we've got two different types. The B cells, remember, they secrete antibodies. That's all a B cell is it's going to be an antibody secreting cell. So we've got three types out of these two main categories. So we've got a CD3 positive cell. It's going to be these. T helper cells will express CD3. Cytotoxic T cells will express CD3. So what else do we have? Let's move on to CD4. What does a CD4 positive cell mean? Well, this means that we're going to bind. Maybe you've heard before CD4 always binds MHC2. MHC2. You may have heard that before. So the MHC2, let's talk about this first. MHC2 is going to be a complex that's going to be found on antigen-presenting cells. So let's say a dendritic cell, for example. It's going to grab stuff from the environment. It's going to grab antigens from the environment, maybe a protein, maybe a saccharide, maybe a sugar. And then what it's going to do is it's going to break it down into fragments. And what it's going to do is it's going to break it down, so it's going to eat it up kind of like Pac-Man. Nom 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 nom. And then it's going to spit it out on this MHC2 process on its surface. So it's going to chop it up. We're going to express parts of that stuff that it ate up onto its surface. So we've got a little bit of antigen here, like a fragment of that antigen that it just ate up, and now it's being expressed. Along comes a CD4 positive cell. It's going to bind. It's going to have co-stimulatory factors that it also must bind. I'm just going on the very basics, but it's going to find this MHC2. It's going to bind. These CD4 cells are going to recognize MHC2. When we say MHC1, what does that mean? Well, all cells except for red blood cells, primarily red blood cells, all cells have MHC class 1. It's going to do the same thing. It's going to snip up stuff. However, this is going to be more intracellular. So instead of finding all these molecules outside the cell, you're going to have all these pieces. Maybe it's rough endoplasmic reticulum. Maybe it's part of the nucleus. Maybe it's a lysosome. Part of whatever is found in the cell. Going to break that up into small little fragments, stick again onto a cell surface marker. This time it'll be MHC1. And we're going to express intracellular stuff. Well, why are we doing this? Well, here we go. We've got extracellular stuff that's going to be expressed by MHC2. Along comes CD4, it's going to bind, it's going to stimulate T helper cells. T helper cells help stuff become active. It's going to help a B cell become active. It's going to help a cytotoxic T cell. So T helper cells don't actually do much themselves. They'll help. So that's why they're aptly named T helper cells. They're going to help stimulate cytotoxic T cells. They're going to help stimulate macrophages. They're going to help stimulate B cells to produce antibody. Let's see if I can do that better. That doesn't look like antibody. Antibody. Okay, so let's go back here. I know I'm kind of going off on tangents, but I do think that this is important to understand. So this extracellular stuff is going to be degraded, and the extracellular stuff will be expressed on MHC2. Along comes a T helper. It's going to say, hey, that extracellular fragment doesn't look normal, so there's something in our extracellular space that's wrong, so therefore we should activate extracellular defense through antibodies or through cytotoxic T cells. Okay, so now we've got MHC class 1. It's going to express intracellular stuff that's found inside that cell. Let's say it's infected with a intracellular microbe, so something that lives inside the cell. It's going to chop that up. It's going to express part of that little molecule there. The body recognizes that this cell is infected because this MHC1 always binds CD8 positive. So when you think MHC1, you think CD8. When you think MHC2, you think CD4. Easiest way to remember this is 4 times 2 is 8. 1 times 8 is 8. So 8 is the golden number. Well, what does this mean? It means CD4 times MHC2 is equal to 8. MHC1 times CD8 is 8. There's only one combination of how you get 8, and that's how you do it. That's how I remember it, at least. So we've got MHC1 and MHC2. I haven't yet said what CD4 positive is, and I did want to give you a little bit of a background before I went to it. CD4 positive cells simply means it's going to be a T helper cell. I kind of alluded to it over here where you have these extracellular fragments or extracellular antigens get degraded by Pac-Man over here, the cell. This is going to be an antigen presenting cell. It's going to present it on its MHC class 2 cell surface marker, and that is going to bind to a CD4 positive cell. That CD4 positive cell, also known as a T helper cell, which will also be expressing CD3 because it's a T cell. So this T helper will have CD4 and it'll have CD3. It's going to bind that MHC2. It's going to say, hey, there's stuff in the extracellular environment, we need to get rid of it. The T helper cell will help activate other stuff to get rid of it. Alright, so a lot of talking about only 2 things. We need to pick this up a little, otherwise this video will be very long. So I did already allude to CD8 positive. What is a CD8 positive? It's going to be a cytotoxic cell. So remember, we have a T cell. It can either be a T helper cell or it can be a cytotoxic T lymphocyte. So it'll be a cytotoxic cell. These are our big boys. If a cell is infected, it can be either eaten up by a macrophage. Macrophages are part of our innate immunity, meaning it'll be there always. We don't have to ramp it up before it's active. So a macrophage will be part of our innate immunity. It can be eaten by a macrophage. It can be killed by a natural killer cell. Natural killer sounds pretty big, important. It's not. If a natural killer cell can't kill it, what will happen is you'll need to recruit a cytotoxic T lymphocyte, also known as a cytotoxic cell. These are like the minor leagues. This is the major leagues right here. If you want to watch football, you don't want to watch, well, maybe you want to watch minor leagues, but I always want to watch NFL instead. So you have the cytotoxic cell, the cytotoxic T lymphocyte. These are the big players. These will kill intracellular organisms. So you'll have a cell here, cell here, cell here. Middle one's going to be infected with red. the microbe, our cytotoxic T lymphocyte will come along, say, kill it. Alright, so that's CD8. CD8 is simply meaning it's going to be found on cytotoxic T cells. Also remember that it should also have CD3 because CD3 simply means it's a T cell. So a cytotoxic T lymphocyte will have CD3 and CD8, while that's over there, our T helper cell will have CD4 positive and CD3 positive, so 4 and 3. Okay, so I've already covered all that. Pretty good detail. Hopefully it's making sense. Next we've got CD28. So we have a CD28 marker. What is that? Well, let's look back. If it'll go back. Okay, we've got this. Remember I said you've got this MHC class 2, you've also got this MHC class 1. Then I also said there are a whole bunch of other things, other factors. Let's see if I can get a different color. Other stuff over here. You've got these co-stimulatory molecules. Well, you've got CD28. And what this is going to be is this will be one of those co-stimulatory markers. So you need the CD28 to be effective. If you don't have a CD28, you're not going to be working. So not only do MHC class 2 plus CD4 need to be matching, you also need to have B7 match with CD28, and this will also be required. So you need one reaction. This is a co-stimulatory, meaning it needs to be activated as well. Somehow B27 cell surface marker is going to match with CD28 cell surface marker. It's going to be co-stimulatory. Alright. Anything you need it for the T cell activation. So notice how I've talked about T cells. I've talked about the basics of the T cell. I've talked about T helper cells. I've talked about cytotoxic T cells. I've talked about co-stimulatory molecules for T cell activation. For that T cell, for this, did I do it? I didn't draw anything out for T cells. For a T cell to become active, for the cytotoxic T cell to be active, it'll need to bind to its MHC1. It'll also need to bind to its co-stimulatory CD28 marker. Alright, so what else do we have? Let's see if I can get a new slide here. So I just talked about T cell stuff. Now let's talk about B cell stuff. B cell stuff. Alright, we've got CD19, we've got CD20, got CD21, all in a row. 19, 20, and 21. This makes it really easy. All this is, there are stuff for like Epstein-Barr virus, not as important right now. I just want to point out this all means B cell. When you see CD19, when you see CD20 and 21, you want to think B cell. B cells are going to secrete the antibodies. B cells secrete antibodies. Yeah, that's about all I want to go into that. Now let's talk about macrophages. B cells are going to be part of your adaptive immunity. Maybe I'll make a video about adaptive versus innate if I haven't already. The adaptive immunity, you're going to have to ramp it up. So you have a piece of antigen here, goes into your body. You're not going to react to it right away. You're going to be affected by that antigen. So let's say this is a mono bug. No, let's say it's a chickenpox bug, because I just got done making a video with chickenpox. I like chickenpox. So we've got chickenpox. Got our patient here. They're going to be infected with chickenpox. Right away, you're going to have some macrophages. You're going to have some neutrophils. You're going to have some NK cells, so some natural killer cells. All this stuff will start trying to get rid of this varicella microbe right away. However, this is going to be your innate. They are innately there. Innate. So they are innately present. Doesn't matter what this is. They'll try and get rid of it if it's not normal. However, it's also weaker. It's not as strong. So then we've got the adaptive. The adaptive immunity. And an adaptive immunity, you're going to see this varicella bug. It's going to go into the body. Your adaptive immunity, unless if they've already seen it before, they're going to say, what? I have no clue what that is. However, it's going to activate B cells. Activate B cells. So if it can kill it via antibodies, you're going to secrete antibodies. Or it's going to recruit T cells, and you're going to need your T helper cells. And then also your cytotoxic T lymphocytes. Remember I said those were really strong and powerful? So you're going to activate your cytotoxic T lymphocytes. Takes time. Takes time. But it's very strong. Strong. Okay. Didn't know this is a spelling bee. Alright. So we've got our adaptive. Ooh, I tried to change color. We've got our adaptive. We've got our innate immunity. So your macrophage. Whoa. Where did I get off to a tangent there? Started talking about macrophages. Okay, so macrophages. If you hear why do I write the D first? CD14, or if you hear CD40. You may see these molecules on others. However, if I hear CD14 or 40, I'm thinking macrophage first. I'm thinking macrophage until proven otherwise, instead of innocent until proven otherwise. You may also see this CD14 maybe like on a dendritic cell or a couple other cell types. What this is going to do is the CD14 is going to bind the lipopolysaccharides and express it there. So the CD14 binds lipopolysaccharides, CD40. I'm thinking macrophage. It just simply means it's a cell surface marker for macrophages. Less important of a function as of CD4 and CD8. Just like CD3 meant it was a T cell, CD14 or CD40, a lot of 4s in here, macrophage. Alright. New slide here. Kind of going to finish up with just a couple other ones. You've got CD16. When you hear CD16, like I said, it can be associated with a whole bunch of stuff. Well, CD14, CD40 were associated with macrophages. CD16 can also be on macrophages. Ooh, phages. It can also be seen on other stuff like NK cell. It can also be seen on neutrophil. Well, this is interesting. Didn't I just say macrophage, neutrophil, NK cell? When this all dealt with your innate immunity. So at age 16 or CD16, you have innate ability to do stuff. Your innate stuff is going to be your macrophages, your neutrophils, your NK cells, stuff that's going to be innately present. So CD16 just simply means it's going to be part of one of these. When I hear CD16, I'm thinking innate. Instead of having to remember 3 of those at a time, I just think innate. Alright, and then lastly we'll end with a CD56 molecule. You're saying what happened to the rest of them? CD1, CD2, I don't care about those. Those are lesser important. I'm just touching on the main ones that I like and the main ones that I think are important. So sorry if I didn't cover one that you wanted me to cover. Maybe I'll make another video on those if I get some requests. However, so what this is going to be, it's going to be adhesion molecule. So neural cell adhesion molecule, and simply meaning it's used for cell adhesion. Thus the name. Where you're going to see these, you're going to see these in NK cells. You're going to see them in neural cells. Also, it's typically found in NK cells and neural cells. However, if a cell is producing CD56, but it is not a natural killer or a neural cell, I'm thinking tumor. So CD56 may be a tumor indicator. So if you're checking different cells and you see CD56, you want to rule out, is it a natural killer cell? Is it a neural cell? If it is, it's normal. However, if it is not a natural killer cell or a neural cell, I'm thinking tumor. So it could be a pathologic marker or a tumor indicator. Let's see, I just want to go back to the first screen, make sure I hit all, yeah. I just wanted to cover the basics. That was my goal. If you have any questions, be sure to ask. Otherwise, I really enjoy hearing comments, whether you liked it, whether you hated it, whether you hate my guts. If you did enjoy this presentation, please click like. It builds my ego up hugely. Otherwise, if you want more great videos, subscribe for more. Thank you very much for paying attention today, and I hope to see you all again.